Hello, everyone. I'm Heino Heyman, Director of Field Metabolomics Sciences at Metabolon. Welcome to episode 7 of our Molecules to Medicine series. Today, we're exploring how metabolomics can augment patient stratification analysis and also provide crucial insights into the mechanism of action and efficacy measurements. In this case study today, we will be taking a closer look at how metabolic profiles can identify the right patient for your therapy while deepening your understanding of how and why the treatment works. In this specific study, it was a combination of using both GWAS and metabolomics. And the interest was to investigate if a predictive response phenotype exists for antihypertensive therapy. As with many drug therapies, antihypertensive drugs struggle with a low responsiveness to therapy, with only 40 to 50% of patients responding adequately to the blood pressure control drugs. The variability of this can only partially be explained by genomics or genes, and thus the need exists for additional measurements to pinpoint the nature of the selective response. So a study was conducted, and the specific aim of the study was to replicate GWAS analysis of the hypertensive effects of bisoprolol as well as losartan in the general study by using the Finnish patients in the live study cohort. So looking specifically at the details of the study, which started out with the GWAS analysis, where they analyzed association of 4 specific SNPs that were associated with atenolol response, and 3 SNPs that were correlating to losartan response in these 927 Finnish live patients. The GWAS analysis in the study pointed to a strong association between a variation at the transcription start site of the aminoacylase 3 gene and the response to a treatment of atenolol. So the SNP RS25144036, which is a variation at the transcription start site of aminoacylase 3 gene variation, ACY3, was this very strong showed this very strong association with blood pressure response. EQTL data in 4 different databases also showed strong association with the SNP with aminoacylase 3 expression being shown to be in several different tissues as well. Looking specifically at metabolomics data, the global metabolomics analysis that followed was performed specifically on 35 subjects from the live cohort. And from these 35 individuals, 175 plasma samples were provided. So they are cross-sectional as well as longitudinal samples. And the result was 600 metabolites were identified across all of these samples on Metabolon's Global Discovery Panel performing untargeted metabolomics. The metabolomics data showed negative correlation between the substrate and products of aminoacylase 3. As can be seen in this diagram, the response to bisoprolol correlated to baseline levels of N-acetylphenylalanine as well as phenylalanine in the general study. So aminoacylase 3, these are specific to aminoacylase 3 in that they are both substrate and end product respectively. So the value that metabolomics provided in this study is the confirmation of the associations of aminoacylase 3 involvement in this unique blood pressure therapeutic response. So the metabolomics data really confirmed and validated the genomics data. But it also identified possible biomarkers in plasma that could predict the antihypertensive response to beta blockers. The plasma concentrations specifically of N-acetylphenylalanine and phenylalanine, which are the ACY aminoacylase related catecholamine synthesis precursors, may predict the antihypertensive response to beta blockers, thus providing support for two pharmacogenomics markers for beta blockers and angiotensin receptor antagonists. So, and when we take a look at these four molecular structures here, when we think about phenylalanine and N-acetylphenylalanine as being the substrate and product, they look very similar to bisoprolol and atenolol in structure. So that can then confirm or give an indication of why there might be a competitive environment to this target protein, and why it would potentially take the natural molecule rather than the drug substance. Finally, this study is also a good example of the power of combining both metabolomics and genomics in the study of multigenic and complex diseases. And thus, having both data sources, genetics and metabolomics, provides for some powerful data for your research. Right. So this brings us to the end of this episode, and thank you for joining us today. The power of metabolomics in patient stratification represents a tremendous opportunity to improve clinical trial success and bring more effective therapies to patients who need them most. I encourage you to explore how Metabolon's expertise can support your precision medicine initiatives. Our team is ready to help you design studies that leverage metabolomics for better patient stratification, and better patient selection, and treatment monitoring. This episode is one piece of our complete Molecules to Medicine series designed to address every stage of the drug development process. Feel free to watch other episodes on your current priorities, whether that's biomarker discovery, translational research, or drug safety assessment. We value your feedback and want to ensure our content meets your needs. Please share any specific topics or challenges you'd like us to address in upcoming Metabolon series. Together we can advance science that brings life-changing medicine to patients. Thank you for your time.